19. We're on bacteria part 2 today. So we're going to talk about today is more ways that we identify prokaryotes. So we know now that they're put into two groups, if you remember, from the eubacteria and the archaea bacteria. So now we're going to look at some more specific things that we use to actually identify them on an individual level, kind of more specific than just putting them into one of those two groups. So they can be identified by kind of 4 different characteristics that we use. First one being the shape, so that's a pretty simple one. We just look at them and look at their shape. The chemical nature of their cell walls, we discussed that one, remember, with peptidoglycan. It's just a molecule that we need to know it is present in eubacteria, not present in archaea bacteria. They can be classified or identified by the way they move and the way they obtain energy. So the first one we're going to look at is shape. So the first one is if the bacteria is in a rod shaped, it is known as a bacilli. So these are going to be some new words, but might just have to memorize these. We'll do some activities to help us remember them. But bacilli is going to describe rod-shaped bacteria or rod-shaped prokaryotes. You can see they give a pretty good example in the picture right there. Then the next one, spherical prokaryotes, are called cocci. An easier way to remember the spherical is just kind of a fancy way to say circular. It's just kind of like a 3-dimensional circle. So circle or cocci, they both begin with a C. So that's our second shape. Our third and final shape is going to be used to identify spiral and corkscrew shaped prokaryotes, and these are called spirilla. So there you can see spirilla as a spiral. Those names kind of sound correctly. So that's how, or sound correctly, sound similar. So that's how we identify them based off of their shape. They can either be spirilla, cocci, or bacilli. Bacilli is rod, cocci is circular or spherical, spiral is spirilla. The next way is by their cell walls, and there's 2 different types of cell walls that are found in our different eubacteria, and we can tell them apart by a method that's called Gram staining. Will help us tell them apart. So within eubacteria, they're gram positive, meaning they have a large amount of peptidoglycan, so they have thick cell walls with large amounts of peptidoglycan. And then there's gram negative bacteria, and they have thinner cell walls inside an outer lipid layer, so they just have kind of, these are the ones, the gram negative would be the ones that have 2 types of cell membranes, whereas the gram positive would be they have just 1 cell membrane and it's a lot thicker. So that is just going to be one way that we can tell them apart within that one group, is just by using this method called Gram staining. I would just want you to know that this is used to identify different types of eubacteria. And then movement, prokaryotes can be identified by whether they move and how they move. So that's pretty simple. There's kind of some more specifics of ways they can move, which we'll talk about later. So they can either be identified whether they move at all and how they move. And our last one is metabolic diversity. So this is just going to be how they obtain energy, is basically all this is going to be, how they kind of take in and break down energy. So they can be divided into 2 main groups, and these are words that we've heard before. Heterotrophs, so a prokaryote can be a heterotroph, meaning they get their energy by consuming organic molecules made by other organisms. So that's basically they can't make their own food, they have to get their food from an outside source. And they can either be autotrophs, which make their own food from inorganic molecules, meaning they can produce their own food like plants can produce their own food via photosynthesis. Autotrophs can produce their own food from some other source other than sunlight, and some can in fact probably use sunlight as well. So heterotrophs, we have 2 different types of heterotrophs here. We have prokaryotes that take in organic molecules for both energy and a supply of carbon, are called chemoheterotrophs. So these names are going to sound pretty confusing, but it's just, if we break it down, you know what the heterotroph part means. Just need to know what the chemo means, meaning that they have to take in their organic molecules for both energy and a supply of carbon. And then there are prokaryotes that will use the sunlight for energy. So again, that's kind of the big difference there. They actually can take in the sun for energy, but they also take in other organic compounds as well for carbon, and those are called photoheterotrophs. So if you remember photosynthesis that plants did, that's taking in the sunlight. Photoheterotrophs have to take in the sun for energy, but they also need to take in another source of food for their source of carbon. So those are our 2 types of heterotrophs. And our next one are autotrophs. So photoautotrophs will use light energy to convert carbon dioxide or CO2 and water to carbon compounds and oxygen. So it's basically, they do basically what a plant does. They get all their energy from the light, and they can kind of just convert it all themselves. So photo, kind of like photosynthesis, autotrophs, that should be an easy one to remember what they do, that they use energy to kind of, from the light, to convert CO2 and water to carbon compounds and oxygen. Then there's chemoautotrophs. These ones are a little bit different. They perform what's known as chemosynthesis, and that means they take in organic carbon molecules from CO2, but they do not require light as the energy. So that's a big one. They do not require light as their source of energy. Do not require light as the source of energy. So photoautotrophs have to have light. Chemoautotrophs, they can make their own food without light. So that's the main difference between those two. Our next one is releasing energy. Again, this kind of still falls under how they kind of make energy. But bacteria need a constant supply of energy, which is released by processes of cellular respiration or fermentation or both. So if you remember cellular respiration, it's just the cells making energy. And so within this, there are three different groups. There can be what are called obligate anaerobes. And if you think of the word obligate, that kind of means you have to do it. So if you're obligate aerobe, sorry I said that wrong, you require a constant supply of oxygen, meaning you're always going to have oxygen. You're obligated to oxygen. And then the next one are bacteria that live without oxygen because they may be killed by it, are called obligate anaerobes. So if you remember, we see the letters AN, that means without oxygen, so they cannot have oxygen. Then our last ones are bacteria that can survive with or without oxygen, are known as facilitative anaerobes. So that means it doesn't matter, they can really do whichever one's convenient for them. So that's it for this video. Let me know if you have any questions.